everyone, welcome to TriRide. This is Pack 365, myself Srisha. So today in this session you're going to learn about the types of immune cells and also the types of immunities. So what are B cells? So B cells, these are the antibody producing cells of the adaptive immune system. So what is the structure of the B cell? So these are the small round lymphocytes. They have the surface immunoglobulin molecules as well as IgD, IgM in the naive B cells. So these surface antibodies react as B cell receptors and also they help the B cells to recognize the antigens. So what are the important markers? CD19 and CD20. So these are the classic B cell markers which are used in the laboratory identification and clinical testing. So CD19. So these are found on almost all the B cells. CD20, these are targeted by the drugs in autoimmune diseases and lymphomas. So what are the functions of the B cells? So they produce the antibodies. So the primary role of the B cell is to make antibodies that neutralize the viruses and toxins, that hopsonize the pathogens, nothing but they help the phagocytes to eat them. They also activate the complement system. So B cells, they act as antigen presenting cells. So B cells, they can take in antigen, they can process it and they can present it on MHC2 system to help with T cells. So the interaction is essential for B cell activation and also class switching. So the other function of B cell is to differentiate into two major types. So after activation, B cell, they can be activated into plasma cells, which are the antibody factories, and also the memory B cells, which last for years, providing immunity after infection or vaccination. So here is a diagram which shows you the B cell activation. So how does the B cell gets activated? The life cycle of an activated B cell. So first, the virus infects a healthy cell. So virus enters and infects a nearby cell. Then the B cell recognizes the antigen. So a naive B cell with surface Ig, it binds to the viral antigen. So then the cytokines which activate the B cell. So the helper T cells releases the cytokines. These cytokines, they fully activate the B cell. Then the activation causes a clonal expansion. Like the activated B cell, what does it do? It divides many times, like producing a clone army of identical B cells, which is called as replication. So then it is differentiated into two types, the plasma cell and the memory B cell. So plasma cells, this produces a large amounts of antibodies. So they are short-lived from days to weeks maximum. Whereas memory B cells, they are long-lived. So they provide the rapid response on second exposure. They also provide the basis of long-term immunity and vaccines. So the plasma cells, they releases the antibody finally. So these antibodies, they bind to the virus or infected cells. They enhances the phagocytosis, which is shown by phagocyte eating the pathogen over here. You can see the red color one here. So this neutralizes the eliminate infection. So this is how the activation of B cell takes place. So next is plasma cells. So what are plasma cells? These are the final fully differentiated form of a B cell. So the structure it is a large cell. It is specialized for a protein production like antibodies production. So it has a clock phase nucleus. Like chromatin arranged in clumps around the nucleus, it resembles a number on a clock, and which is a classic plasma cell feature. So this plasma cells, it has a basophilic cytoplasm. So it appears blue on staining due to abundant ribosomes like RNA, ribose nucleic acid. So plasma cells, they also has this abundant rough endoplasmic reticulum. So nothing but RER. So plasma cells, they need massive RER for antibody synthesis. So this diagram shows the RER mitochondria as well as Golgi nucleus. So it also has a well-developed Golgi apparatus. So what does this Golgi apparatus do? It helps to modify, package and secrete the antibodies. So this is often, it appears as a perinuclear HOF, a clear zone nucleus. So it shows, here is a diagram which shows your typical plasma cell. So it has antibodies which are secreted from the cell, a nucleus which shows your clock phase appearance, and Golgi apparatus, it is a processing center, mitochondria, energy production, and then rough endoplasmic reticulum, antibody synthesis. So overall, it visualizes how plasma cells are the antibody factories equipped with organelles needed for the producing large quantities of immunoglobulins. So this is about the B cell and plasma cell. So next, specialized immune cells. So besides the classic B cells, T helper cells and cytotoxic T cells, the immune system has other specialized cells that bridge the immunity between both the innate and adaptive systems. So these cells respond faster than the typical T cells, and they often recognize the non-peptoid antigens like lipids, metabolites, and stress molecules. So what is the first type? NKT cells. So natural killer T cells. So what are they? These are the hybrid immune cells. So they have characteristics of the T cells, TCR expression. So they behave like NK cells which kill rapidly plus release of the cytokines. So think of them, they act as a fast acting T cells with NK like power. So what are the key features of NKT cells? They recognize lipids, not peptides. So unlike typical T cells that bind the peptide antigens on the MHC molecules, this NKT cells, they recognize the lipid antigens presented by CD1D marker. And they also involved in the rapid cytokine production. So they releases a large amounts of interferon gamma which stimulates macrophages and antiviral, and also the interleukin IL-4 which helps the antibody switching. So they influence both innate and adaptive immunity. And these NKT cells, they act early during the infection. So they activate quickly, almost like a innate cells. So here there is a NKT cell over here at the left side. So the receptor binds alpha galcer, that is alpha galactosylglyceramide, so or the other lipids. So these lipids, they are presented to NKT cells via CD1D marker, right? So mouse MO. So mouse and human, that is V alpha and V gamma chains, they are shown, which are examples of a TCR usage. And right side, it is mate cell, nothing but a mucosal associated invariant T cell. So mate cells recognize the vitamin B2, that is riboflavin metabolites, which are presented by MR1. So these cells, they are common in liver, lungs, and gut mucosa. They also behave like the innate T cells. So next is gamma and delta. So gamma delta T cells. So these are the unconventional T cells whose TCR is made up of gamma and delta chains, not alpha and beta like normal T cells. So what are the key characteristics? They are formed in mucosa, skin, and gut. So they act as a frontline defense. So these are the early responders to stress that detect the tissue damage, heat shock proteins, lipids, and microbial metabolites. So they do not always require antigen presentation through MHC. They even link innate and adaptive immunity. So they act like quickly like innate cells, but also have TCRs like adaptive cells. So what are the important subsets in humans? V delta 1, which is found in mucosa. And here is a mouse gamma delta T cells and human gamma delta T cells. So mouse gamma delta T cells, these are the various subsets, V gamma 5, V gamma 4, and V gamma 6, etc. So they have these specialized functions such as tissue repair and inflammation. Whereas in human gamma delta T cells, the diagram shows V delta 1 and V delta 2, V gamma 9, where blood recognizes the phosphoantigens. So BTN2A1 or BTN3A1, this is shown as a molecule which is needed for gamma delta T cell activation. So they usually respond to the stress related ligands instead of the peptides. And the third type is innate lymphoid cells. So these are the innate versions of the helper T cells. So they mirror TH1, TH2, TH7 functions, but they do not have the TCRs. They act immediately in the infection, tissue repair, and inflammation. So what are the types of innate lymphoid cells? ILC1, which produces interferon gamma. ILC2, which produces IL5, interleukin 5 and interleukin 13. So these are important in allergy and asthma. Whereas ILC3, it produces IL17 and IL22. So these are important for gut immunity, mucosal protection, and as well as maintaining the microbiome balance. So this is about the immune cells. So next, coming to types of immunity. So what are the types of immunity? First of all, what is immunity? Immunity is nothing but it's the ability of the body to fight against the infections and protect itself from the diseases. So the immune system, it is broadly divided into two types: innate immunity, which is a natural and non-specific, and adaptive immunity, which is acquired and specific. So this adaptive immunity, it is again divided into active immunity and passive immunity. So let's go into brief description about these types of immunity. The first one is innate immunity. So innate immunity is the first line of defense. So it protects the body from birth and it responds quickly to any foreign invader. So why it is called as innate? Because you are born with it. It does not require prior exposure to a pathogen. So why is it called non-specific? Because it does not target any particular pathogen. It responds the same way to the bacteria, viruses, fungi, parasites, and toxins. So now what are the characteristics of this innate immunity? It is present at birth. Like you do not need any previous infections or vaccines for it to work. For example, like skin, stomach acid, and phagocytes. Next character is immediate response. So this acts within minutes to hours. For example, like fever, inflammation, and neutrophil response. And third one is it is non-specific. So it is one general response for all the pathogens. For example, like NK cells kill any abnormal cell without identifying the organism. And innate immunity does not have memory. So it does not improve with the repeated exposure. Whether it's the first time or the 10th time, the response is the same, and it provides early protection. So the innate immunity protects you until the adaptive immunity becomes active, which takes days. Right, adaptive immunity, it takes days. So what are the components of the innate immunity? Physical barriers. So physical and mechanical barriers, chemical barriers, cellular components, and physiological responses. So these are all the components of the innate immunity. They are divided into four main groups. So physical barriers. So the structures physically block the pathogens from entering the body. How? Coming to skin, it is a tough, thick and waterproof barrier. Waterproof layer. So it contains keratin that prevents the microbes from penetrating. So skin has natural oils and slightly acidic pH which further inhibit the microbes. So then mucous membranes. What does this mucous membranes do? They line the respiratory, digestive and reproductive tract. They produce as the mucus. So what is the role of mucus over here? It traps dust, bacteria and viruses, nothing but microorganisms, right? So next is cilia. So cilia are hair-like structures in the respiratory tract. So they move rhythmically to sweep the trapped microbes upward to be coughed out or swallowed. So it sweeps the pathogens away out of the body. And tears and saliva, this washes the microbes away. They contain tears and saliva. They contain an enzyme called as lysozyme. This is which enzyme that destroys both the bacterial cell walls. Okay. So this is about the physical barriers. So next is chemical barriers. So chemical barriers, these are the chemicals which kill or inhibit the pathogens like stomach acid, hydrochloric acid. So HCl, it is extremely acidic. It has pH 1 to 2. So it kills many swallowed pathogens instantly. It also has a lysozyme, tears and saliva. So this lysozyme, it is found in tears, saliva, sweat and mucus. It breaks the bacterial cell walls and the bacteria gets bursted out. And antimicrobial peptides like defensins. So defenses, these are the small proteins that punch the holes in the microbial membranes. Okay, and sweat. So sweat is slightly acidic which has low pH. This sweat, it inhibits the growth of the bacteria. So this is how the chemical barriers work in the innate immunity. And the third one is cellular defenses. So cellular defenses, the cells are that immediately attack the invaders. So what are the main innate immune cells? Neutrophils. The neutrophils, these are the first responders which kill bacteria by phagocytosis. And next is macrophages. These are the large phagocytes which engulf pathogens plus dead cells. And dendritic cells, they capture the antigens and they activate the adaptive immune system. And natural killer cells. So these are the cells which destroy virus infected and cancer cells. Mast cells. So they release histamine. So this starts the inflammation. Eosinophils, they fight parasites and allergies. Whereas basophils, they releases the histamine and heparin in the blood. So these are the cells that immediately attack the invaders. So these are the immune cells of this immune system. So here is a diagram which shows you how skin responds when a pathogen enters. So it shows you both the inflammatory response as well as the immune cell recruitment. So first, this is the epidermis, the top layer. So keratinocytes, they detect the pathogens over here and releases cytokines, the warning signals. So next, the activated Langerhans cells. So they capture the antigens and they travel to the lymph nodes to activate the T cells. So next, the middle layer is dermis layer. So the activated fibroblast, what does it do? It releases more cytokines. So this signals, they causes the vasodilation where the blood vessels get widened and it increases the permeability. The immune cells, they can move out of the blood. And it causes a redness, heat and swelling, which is the classic inflammation signs. And then immune cell recruitment takes place. Innate effector cell recruitment. So the chemokines, they attract the neutrophils, which arrive first, monocytes, they become the macrophages, and NK cells, they become kill infected cells. So then movement to the lymph nodes. So antigen presenting cells like dendritic cells, they migrate to nearby lymph nodes. So here they activate the adaptive immunity. So T cells to B cells, creating antibodies as well as memory cells. So the overall process in the diagram is the pathogen enters the skin, Langerhans cell capture the antigen, then keratinocytes, they releases the cytokines which produces the inflammation. So the immune cells rush to site, then the dendritic cells go to lymph nodes, activate T and B cells. So this shows how the innate immunity begins its defense and then activates the adaptive immunity, which takes more time. So this is how about the innate immunity works. So next is adaptive immunity. So what is adaptive immunity? It is second line of defense. So unlike innate immunity, it is highly specific and it develops after exposure to a pathogen. So adaptive is also called as acquired or specific immunity. So what are the characteristics of adaptive immunity? It develops during life. You are not born with adaptive immunity. So you acquire it. So when you get infections or when you get vaccines, you acquire it. So it also has slower initial response. Like it takes days to weeks to the first time a pathogen enters. So the body needs time to build antibodies and activate the T cells. So it is highly specific. Each pathogen gets its own custom-made response and it has memory. On second exposure, the response is very fast. So pathogen is destroyed before causing the disease. This is why you usually get chickenpox only once. And then it also has a long-term protection. Vaccination works because adaptive immunity creates both the memory B cells as well as memory T cells, right? So there are two branches of adaptive immunity again. The first is humoral immunity, which is antibody-mediated immunity, which consists of B cells and antibodies. And the other one is cell-mediated immunity, which has T cells. So the first one is humoral immunity. So antibody-mediated immunity. It is mediated by lymphocytes, B lymphocytes. So how it works? So step by step. And here is a diagram which shows you the humoral immunity left side and the cell-mediated immunity at the right side. So just have a look on this. So the humoral immunity, the B lymphocyte recognizing the antigen. So then the B cell activation happens and after B cell activated, it divides and becomes into both plasma cells and memory B cells. So then the antibodies neutralizes the pathogen. So the main concept of humoral immunity is it fights invaders outside the body. Whereas cell-mediated immunity, it is mediated by T lymphocytes, T cells, especially helper T cells and cytotoxic T cells. So how it works? A pathogen invades and infects a cell. The infected cell or a dendritic cell, it displays the antigen to the T cells. So once T cells become activated, it forms into helper T cells and cytotoxic T cells. So this is a main defense against the viruses, intracellular bacteria as well as the cancer cell. So humoral immunity. So what is humoral immunity? I already said you that it is mediated by B lymphocytes. So how it works? So how does the process of humoral immunity takes place? Humoral itself, it is the immunity found in body fluids like blood, lymph, mainly through the antibodies. So the first step is the B cells the B cells recognize the antigens. So the B cells have receptors, BCRs, that bind to specific antigens. So for example, a B cell may recognize the bacterial surface protein. So the second step is it differentiate into plasma cell. So once it get activated, it is often with the help from helper T cells. So B cells turn into plasma cells. So the antibody factors of your body. So then the plasma cells produces the antibodies. So these antibodies are circulated in the blood and they attach to the antigens. So what does the antibodies do? Antibodies, they help to eliminate the pathogens in several ways, like neutralization, so block toxins or viruses from binding to cells. Opsonization, they coat the microbes so phagocytes can easily eat them. And activation of the complement system, it leads to destruction of the bacteria. So then agglutination, it clumps the antigens together to make the removal easier. So this humoral immunity, it is most effective when dealing with the pathogens outside the cells. So because antibodies cannot enter the infected cells, right? So extracellular bacteria, for example, streptococcus and staphylococcus. Coming to toxins, for example, tetanus toxin and diphtheria toxin. So viruses before entering cells. So once viruses enter the cells, so antibodies, they cannot reach them. So this is when the cell-mediated immunity takes over. So this is how the humoral immunity works. So next, coming to cell-mediated immunity. Now this is T-cell mediated. Humoral is antibody mediated, whereas cell-mediated immunity is T-cell mediated. So this arm of the immune system, it deals with the pathogens inside the cells and with the abnormal cells such as the cancer cells. So they involve the T lymphocytes, like T cells originate from the bone marrow, but they mature in the thymus organ. So where each develops specific receptors to recognize antigens presented on the MHC molecules. So what are the types of T cells and what they do? Helper T cells, CD4+. So they are the commanders of the immune system. So their role is to activate B cells to produce the antibodies. They activate the macrophages to enhance the phagocytosis, and also they releases the cytokines which coordinate other immune cells. So for example, like CD4+ cells, they help the macrophages to kill tuberculosis bacteria inside them. Okay. So next is cytotoxic T cells. So they are the killer T cells. Their job is to destroy the infected or abnormal cells. They releases the perforin, which creates pores in the infected cells. They releases the granzymes, where it enter the pores and trigger the apoptosis, nothing but a cell suicide. And this is effective against the virus infected cells, intracellular bacteria like listeria, cancer cells, and as well as the transplanted tissue. So now what are the regulatory T cells? So they control and they calm down the immune response. So they prevent the autoimmune diseases by stopping the immune system from attacking the body's own tissues. This is about the cell-mediated immunity and the types of T cells. So next, active immunity. So what is active immunity? It happens when your own immune system is stimulated to produce the antibodies and memory cells after exposure to an antigen. So in simple words, your body fights, learns and remembers the germ, right? So what happens next time the germ comes? Your body responds faster and stronger. So in the same way, the active immunity works. So what are the characteristics of active immunity? This features helps to differentiate active immunity from the passive immunity. It develops very slowly. So like this takes days to weeks to develop. So this time is needed for antigen recognition, activation of B and T cells, production of antibodies, and formation of memory cells. And also it has a long-lasting protection. So it produces the memory B cells and memory T cells. On re-exposure, the immune system responds faster, like a secondary response. So this can last years or lifelong. And as well as this active immunity is highly specific. So the immune response targets a particular antigen. So what is the example? So antibodies made against measles. They won't protect against from dengue, right? So each immune response targets a particular antigen. So there are types of active immunity: natural active immunity, artificial active immunity. So what is natural active immunity? So it occurs when you get infected naturally. So if a person gets chickenpox, so the immune system produces antibodies, memory cells. So this gives a lifelong immunity. So why this works? Because the infection exposes the body to the full antigen. So natural exposure leads to a strong specific immune response. And the second type of active immunity is artificial one. So artificial active immunity, nothing but it occurs through the vaccination. So how does it work? Vaccines introduce, weaken, kill or parts of the microbes. So these are the safe versions of the antigens. So the immune system reacts and it forms the antibodies, memory cells. So the person becomes protected without getting the disease. Examples like MMR vaccine, hepatitis B vaccine and COVID-19 vaccine. And here is a diagram which shows you the both active immunization and passive immunization. So this means your immune system is activated and produces its own antibodies. So the natural active immunity and artificial active immunity. So natural active immunity, what happens? It happens after the natural infection. For example, like measles and chickenpox, there is no medical intervention. Whereas artificial, it happens through vaccination. So these vaccinations may include inactivated microorganisms, live attenuated organisms and DNA vaccines. It also consists of the recombinant antigens like subunits, toxides and epitopes. And also this artificial vaccination, it has recombinant vectors which are bacterial or viral vectors. So what is the purpose of this artificial active immunity? It stimulates the immune system without causing a severe disease. And then passive immunization. So here is the person receives ready-made antibodies. So there is no memory is formed. So the natural passive immunity. So antibodies pass from mother to the baby through placenta, through IgG, immunoglobulin and through breast milk IgA. And artificial passive immunity it is given by injection of the antibodies. For example, anti-rabies immunoglobulin, anti-tetanus immunoglobulin and anti-venom for snake bites. These all comes under artificial passive immunity. So this is used for intermediate protection when waiting for active immunity is too slow. Okay. So passive immunity. So this is a type of immunity where the person receives a ready-made antibodies instead of producing them. So you borrow the antibodies from the another source. So your immune system does not make them. So this provides a quick protection, but it does not last long. So what are the characteristics of the passive immunity? Immediate protection. So since antibodies are already prepared, the body does not need time to produce them. So these are useful when quick action is needed to neutralize a toxin or virus. So it has a short-lived. It is short-lived and there will be no memory. There will be no memory B cells or no T cells are formed. So protection lasts days to weeks, not months or years. So once the injected antibodies are broken down, the protection ends. So it is very important in emergencies because this is used when the infection is fast and dangerous. So the person has no time to develop the active immunity. Vaccination takes too long to start the working. And there are types of passive immunity, the natural one and the artificial one. So the natural passive immunity is this type of occurs naturally from mother to baby. How? Through placenta, through IgG antibodies. So during pregnancy, the IgG antibodies, it crosses the placenta. So it protects a newborn during the first 3 to 6 months. So the example protection like from measles, tetanus and diphtheria. And also through breast milk with IgA antibody. So breast milk, especially colostrum, which is rich in IgA antibody, It protects the baby's gut, throat and lungs. So IgA guards against the diarrhea, respiratory infections and gastrointestinal infections. So this is why the breastfeeding is considered extremely important for a newborn immunity. So this is about the natural passive immunity. So next is artificial passive immunity. So this type occurs when ready-made antibodies are injected into a person. So what are the sources of the artificial passive immunity? Human immunoglobulin, antiserum, antivenom from animals like horses. So now what are the examples of this artificial passive immunity? Anti-rabies serum or anti-rabies immunoglobulin. So this is given immediately after a dog bite to prevent rabies until the vaccine starts working. Then anti-tetanus immunoglobulin. This is given when a person has a dirty wound and it is not vaccinated. So next is anti-venom for snake bites like scorpion stings and RSV monoclonal antibody. It is given to the premature babies to protect from the RSV virus, respiratory syncytial virus. So this provides fast but temporary protection. So this is about the artificial passive immunity. So this is for today session. I hope this session has helped you. So in the next session, you will be able to learn the physiology of the immune system. Thank you.